present was recently published just this month. So I'm happy to have that good fortune as well. So let me start by saying that I think most of you would recognize that the retina is an accessible part of the CNS, and it's an accessible part because it's relatively simple and because we know much about all the different cell types that occupy very stereotypic positions within each of its layers. I hope you can see my cursor. So each of the cell types occupies distinct locations, has very stereotypic morphologies, so they're easily identifiable, okay? And so this feature of this accessible part of the CNS has been taken advantage of, and we take advantage of studying the retina in the zebrafish, largely because it's a vertebrate, of course, as you probably know, and has a structure, the retinal structure is very much like ours. It's a genetically and optically accessible model organism. And of course, we take advantage of the fact that the retina and the organism develop rather rapidly. Just to hit home that point about the rapidness of development, on the first day post-fertilization in zebrafish, you wouldn't recognize the laminated structure that I showed you diagrammatically earlier, just because that here the retina is simply proliferating cells. But just 2 days later, I hope you can appreciate that the retina has converted now, if you like, to a laminated structure. And if I were to superimpose the schematic cell types, those are the positions they would be occupying. So to compare with mouse, this time window of 2 days would be about 2 weeks in a mouse. So this is really, really rapid. And we take advantage of this because we do in vivo live time-lapse imaging using promoters, transgenic constructs, essentially, where promoter elements drive the expression of reporters. And here I hope you could see an in vivo confocal stack through the zebrafish eye, leading to mosaic labeling that we like to take advantage of because we can follow individual cells, their behaviors when they undergo division and when they get to their appropriate definitive locations, okay? So the story I'm going to tell you today is about a specific type of interneuron. Interneurons in the zebrafish retina exist here in the inner nuclear layer, and excitatory and inhibitory interneurons are very easily identifiable. So the specific interneuron we're going to concentrate on are bipolar cells. Now in zebrafish, not necessarily in the mouse, but in zebrafish, the vast majority of bipolar cells express visual system homeobox 1, this is VSX1, a transcription factor. And these VSX1 expressing bipolar cells are generated by terminally dividing progenitors that also express the same transcription factor and that are essentially committed to their exclusive generation. Okay, so this was shown a few years ago and we were able to confirm it. But what is interesting is if you look at a stable transgenic line, VSX1 GFP, that was not made by us, but very well established, you will notice not only bipolar cells being labeled, but also amacrine cells. These are inhibitory interneurons also located in the inner nuclear layer. So we were intrigued by what was going on. Why was it that amacrine cells were labeled in this line? Could it have been just erroneous, or was there some kind of lineage relationship? So we were fortunate in having tools generated by Rachel Wong's lab, specifically 2 postdocs of hers, Sachihiro Suzuki and Takeshi Yoshimatsu, had made constructs and stable lines where VSX1 promoter elements, instead of driving the expression of reporter genes like YFP directly, they utilized the GAL4 UAS system. This is a binary system that's very useful to us, borrowed from Drosophila, not only because you can express multiple transgenes, but you can also express not just reporters, but actuators as well. So we were very fortunate to have this long before it was published. And as you can see here, I hope in yellow are bipolar cells being driven by VSX1 promoter elements. For unknown reasons, this particular line that they called Q32, line 32 from the lab, is a subset line of VSX1. In other words, not all VSX1 cells are labeled, but we thought we could take advantage of that because sparseness of labeling really helps to follow cells much more easily than if many cells are labeled. So in work that I'm not going to show you now, we were able to confirm that Q32, remember, which is a subset of VSX1, also produced these terminal symmetric divisions in which two bipolar cells were generated. And I might switch between Q32 and VSX1 interchangeably throughout the talk. Please forgive me for that, but it's really the same thing because Q32 is simply a subset. Okay, so we were able to confirm that, but excitingly, we were also able to see mitotic divisions that generated bipolar cells and amacrine cells, which I hope you will notice now, with the cell losing its apically directed process and assuming the morphology of an amacrine cell. Here is the time-lapse panel, essentially in a static image that you saw earlier. I hope you're convinced, based at least on morphology, that the blue labeled cell is a bipolar cell and the orange an amacrine cell. Okay. Now we know that these amacrine cells are bona fide amacrine cells, not just based on their morphology and their location, but also on the expression of known amacrine cell markers. In this case, PTF1A GFP, this is a transgenic marker. And it's not just a small population of the Q32 progenitors that divided to generate these cells. So almost 50% of them. So it was a substantial number. So we decided to really investigate what the molecular mechanisms were that underlie these terminal asymmetric divisions. And not surprisingly, perhaps, we turned to looking at notch signaling because this system has been implicated in binary cell fate across vertebrate species in invertebrates in determining binary cell fates. So as a reminder, for those of you who may not know, Notch is a transmembrane receptor that interacts with different ligands on neighboring cells, after which a cleavage event occurs where the Notch intracellular domain goes into the nucleus and together with a complex essentially initiates target gene activation. Now this particular complex is being taken advantage of by several Notch reporter transgenic lines that are around in the zebrafish world, and one of them, for example, expresses mCherry whenever Notch signaling is active. So we essentially crossed this line to our Q32 line and asked whether there's any kind of asymmetric expression of Notch signaling in the amacrine versus bipolar cells. And what we found was that indeed the amacrine cell expressed higher levels of Notch. Now, because we do time-lapse imaging, we were actually able to follow the divisions in combination with Notch sensor. And we were able to see that during mitosis or at mitosis, Notch sensor levels are very low, and it only increases several hours post-mitosis and is much higher, as would be expected from what I just showed you, in the amacrine cell compared to the bipolar cell. So we did the obvious things. Gain and loss of function experiments. For the loss of function, we could not do it genetically, or we were unsuccessful doing it genetically. So we used a pharmacological approach using DAPT, which is a gamma secretase inhibitor, to see what the phenotype would be. And as expected, we saw fewer amacrine cells in the VSX1 line compared to the wild type. The opposite in terms of gain of function, we could do, fortunately, genetically, and we were able to overexpress the NICD, this Notch intracellular domain, exclusively in the Q32 GAL4 line, and could show, and I hope you can see, that these orange dotted cells are an excess of amacrine cells when NICD is overexpressed. This is quantified here, so there's way more than doubling of the number of amacrine cells in the Q32 lineage compared to wild type. How do these arise? Essentially, they arise by divisions that don't normally occur. So for example, when you overexpress Notch, or NICD rather, we saw the emergence of 2 amacrine cells being generated by a division, which is something we never saw in the wild type, shown here in a static panel. And when we quantify these kinds of divisions, you can clearly see that the absence of such symmetric amacrine divisions, which don't occur in the wild type, certainly occupy a very large proportion of all divisions following NICD overexpression. But this could not account, these divisions we were puzzled by, because they could not account for the vast numbers of amacrine cells we were seeing. And it might just be a coincidence, but we think not. We think that the transformation of nascent bipolar cells into amacrine cells may account for the increased numbers of amacrine cells we saw following Notch expression. And so here again is just a time-lapse panel of what you just saw in a movie. And what we see effectively is bipolar cells transforming their shape and then acquiring an amacrine cell morphology. What I don't show you here is that these amacrine cells start to express amacrine cell markers. And in fact, amacrine cell markers start being expressed already when the cell is still a bipolar cell, at least based on its morphology. So we did, of course, look at marker expression in these Q32 cells. And I know this is a busy graph, but I'll point you out to 2 things. First, when we looked at bipolar cells or amacrine cells in the wild type situation or following NICD overexpression, what we started to see, as I just mentioned, was the expression of PTF1A, this amacrine cell markers in bipolar cells, and the fact that cells were now starting to express, specifically amacrine cells, were starting to express GFAP. Now, I'm sure you're familiar with GFAP as an astrocytic marker, and it is. In the zebrafish retina, it labels Muller glial cells, but it's also expressed in progenitor cells in the zebrafish retina. So we refer to this as an immaturity marker because I can tell you, and I don't show you here, but overexpression of NICD does not induce excess proliferation. So that's not what we saw. So this is a recap of what I already told you, the excess numbers of amacrine cells following NICD overexpression. What about if we overexpress PTF1A? Oops. Remember I told you about the transcription factor PTF1A being a marker of amacrine cells, but it's actually also a transcription factor that is necessary and sufficient for driving amacrine cell fate. We do see an increase in the number of amacrine cells, as you can see here, but it's moderate compared to the NICD overexpression. When we combine both, in other words, both NICD and PTF1A, we see a dramatic increase in the number of amacrine cells in this Q32 lineage. And here it is, the dramatic increase quantified. And of course, because we can follow, again, these divisions and ask how these, what modes of division contribute to this excess number of amacrine cells, I hope you can appreciate that following the overexpression of both PTF1A and NICD, it seems that divisions are pushed towards generating more amacrine cells, courtesy of symmetric amacrine divisions. Okay, so the story essentially that we began, or the model we began to develop, is that this re-specification of cell fate was happening post-mitotically, that VSX1 progenitors do produce 2 VSX1 expressing bipolar cells, but in about 40-something% of the cases, one of the bipolar cells starts expressing higher Notch signaling. We don't quite know why. We did look at interactors of Notch, and such as NUM, for example, and we looked at orientation of division as something that could occur at the progenitor level and could dictate downstream events in the post-mitotic cells. But we found no such correlation or no asymmetric distribution of a notch antagonist like NUM. And so we still don't know what causes increased notch signaling in one of the bipolar cells, but when this occurs in the wild type, in a physiological state, we think this makes the cell plastic. And I'm being very hand-wavy here. I don't quite know what that means, other than to say that the cell is more malleable in terms of the kinds of fates it can adopt. And PTF1A is overexpressed in that cell, whether there's a direct link, whether notch instructs or binds to PTF1A, we don't know. But this is what we think happens, and this is what we think instructs the amacrine cell fate. So it's not notch instructing the amacrine cell fate directly, it's through PTF1A, so in concert with PTF1A. So if our prediction that notch signaling is causing plasticity in the post-mitotic nascent cell is correct, then we decided to test this hypothesis essentially by doing the following crazy experiment: overexpressing notch using our NICD transgenic line, obliterating PTF1A using a morpholino, and overexpressing another transcription factor to drive a completely different cell fate. And we chose atonal 7 to drive the ganglion cell fate. And the reason we chose this is because this cell type is generated long before bipolar cells or amacrine cells are generated. And essentially, it's well known as a driver of this particular cell fate, and it's completely different to the VSX1 lineage. We were able to get what we call ganglion cell-like cells. So it was not a terribly successful experiment. So the number of times we got such cells was very low, but we were encouraged by the fact that we were able to generate ganglion cells that seem to have ganglion cell-like cells that seem to have features. So they expressed molecular markers like ILO2B. They had axons leaving the retina, and on occasion we were able to see axons even in the optic chiasm. They had dendrites that were not always directed towards the appropriate synaptic partners, but nevertheless they had some of the features and characteristics of ganglion cells. So, and on occasion, we were even able to time-lapse their formation. And here you can see an axon forming in this migrating cell. So in summary, I think, or I hope we were able to convince you that the VSX1 lineage is not exclusively dedicated to bipolar cell production, that re-specification occurs following division in the post-mitotic cell, but this is a very young nascent post-mitotic cell, that notch signaling is a key part of it. It acts in concert with PTF1A to instruct the amacrine cell fate in the VSX1 lineage. And we think that notch is playing a dual role. It's conferring a degree of plasticity to these nascent cells and likely activating a transcription factor cascade. In this particular case, it would be PTF1A. And we think, and this is certainly just conjecture, this kind of plasticity that's exhibited by newborn VSX1 cells might provide a mechanism for regulating ratios of excitatory and inhibitory neurons in emerging circuits, like they are in the developing retina. And with that, I'll acknowledge the two people who actually did the work, Peter Enger and Eleni Petridou, both co-first authors on the paper that was just published. I would also like to acknowledge the line that was given to us that allowed much of this work to be done, generously shared by Rachel Wong and her postdocs. And of course, the DFG, the German Research Foundation, via these particular grants through which we could get funded. And thank you for your attention.